All right. Okay, for today's lab, I was going to do the pre-recording yesterday, as I told those that were attended the lecture this morning. But then when I went over it, I realized, wait a minute, this is a lab. I definitely need to meet with y'all. So it'll be asynchronous most of the time, but I definitely need to meet with y'all here. So today's lab 5 is membrane transport. Okay, it's associated with chapter 7 in lecture. So if you haven't listened to the video and gone through your notes in chapter 7 in lecture, it doesn't matter how I say it, what I say. This lab is not going to make much sense, okay? So keep that in mind, okay? So these labs, several, or at least half of them at least, match up pretty well with the lecture. So if you have trouble understanding this lab, go back to chapter 7, go through your notes, go through the PowerPoint, listen to the video, look at the web links. I provided some web links here too to help you out, okay? Now, the purpose of this lab, to gain an understanding on how the passive process of membrane transport diffusion works, and to gain an understanding on how a special type of diffusion called osmosis also works. So that's the 2 points of focus. So the way I've arranged the PowerPoint, there are parts of the PowerPoint that should match up with your pre-lab and your lab procedures. There's one figure in particular that I believe is given on page 1 or 2 of your lab protocol that I have inserted at a different point on the PowerPoint. So I'll point that out as I go, okay? But basically the way I try to arrange the PowerPoint, as I'll show you, is to help you with part of the pre-lab. Now, objectives, I forgot to get rid of why. That's the old way. This is actually everything you're supposed to learn. I'm going to get rid of that second slide and add this. But the overall objectives: define diffusion, osmosis, simple diffusion, facilitate diffusion, active transport, distinguish between a model membrane, dialysis tubing, biological membrane, identify the factors that affect the rate of diffusion. There are 3 that you're going to test in this lab, that are tested in this lab. Distinguish between movement and net movement in molecules. This is one of the areas I want to talk about, so I'll pull up the PDF version of the protocol and point some things out to help you out. And distinguish between isotonic, hypotonic, and hypertonic solutions, and also explain what happens when cells are placed in each of these types. I'll explain that also in a sec. Okay, so for diffusion, just like with the lecture, I'm giving you the same definition, okay? It says here all atoms and molecules are in constant random motion regardless of conditions. The movement that results from this constant motion is diffusion. So it's this next line here. This is a definition that I mainly want you to focus on, okay, to help you out, because that's what will help you out best for the lab. First part is mainly from the department that wrote it up, but this works out better. Diffusion focuses on the movement of solids from a high concentration to a low concentration of solids until evenly distributed. Now, this is a figure that I used during the lecture. Keep in mind, when I start using the term solute and solution, you have to remember, what is a solution? A solution consists of a solvent and dissolved solids. What is a solvent in this class always? Water, as shown here. What is a solute? Well, any compound, okay, that can be dissolved by what? Water, or mixed with water in the case of ions. In this case, today's lab, it's compounds. Now, they're showing you dropping a sugar cube in to this beaker of water. What's going to be created passively, okay, is simply a sugar solution. Now, they stain the sugar cube, or the molecules of sugar that make up the cube, purple, so we can track diffusion, or the movement. Once they drop that sugar cube at the bottom of the beaker, we now have an area of high concentration. And you can see a little halo around it. This halo represents the molecules that are diffusing or dissolving, being dissolved into solution. But this area where you see a very dark purple color, and then this area where you see the halo of a light color, and then you see nothing, well, what that's representing is what we call the concentration gradient. Okay? Diffusion is a passive process. It doesn't require energy. Okay, it may or may not require a membrane protein known as a channel or carrier protein. Okay, so what's the driving factor? It's a concentration gradient. We have to have an area of high solute concentration next to area of decreasing or low solute concentration. And as long as we have those 2 areas from high to low concentration present, we have a concentration gradient and diffusion will occur. Well, eventually what happens is, the reason why we stain the sugar molecules is so we can see them diffusing from a high to a low concentration until they are evenly distributed in the solution. But in the sciences, instead of saying until evenly distributed in the solutions, in the sciences, we say until equilibrium. Well, the reason why they colored the sugar cube also is so you can see when we've reached equilibrium. At equilibrium, throughout this sugar solution, we have an equal concentration, okay, of sugar per water throughout this. Therefore, we no longer have a concentration gradient. When we are at equilibrium, we no longer have a concentration gradient. You can see the solute concentration is evenly distributed throughout. Except when you apply diffusion to us, the living body, and our cells, you have to factor in the membrane, you have to factor in temperatures and things of that nature. Therefore, part of the experiment, the first part of the experiment, okay, focuses on diffusion and factors that can affect diffusion. So if you look here, the rate or speed of diffusion depends on the atom or molecule size, temperature, and medium through which the molecules are moving. The medium, if it were in our body, would be the cell membrane or plasma membrane. In today's lab, the medium will be either a gel called agar, which is a seaweed extract, sometimes called agarose gel, or it'll be what we call dialysis tubing. Okay, both of those materials will serve as our medium or as semi-permeable membrane. Okay, in the first part of the lab experiment, okay, it tells you you're going to use two different dyes. You're going to add them to a medium made up of agar, and you're going to have three different plates. One of the plates, the amount of agar used to create the medium is much higher than the other two, and I'll point that out. Okay, but you're also, and what's going to happen is the two different dyes you're going to use, well, the reason why you're using two different dyes is because the molecules that make up the dye, the sizes aren't the same. If you have your mic on, can you turn it off, please? So you can see here, size can affect the rate of diffusion. In addition to that, temperature. So also in the first part of the lab procedure, can you check your mic, please, and turn your mic off, please? Can all of y'all check your mic? All right, we're going to do the best we can then here. All right, everybody, turn off your mic if you have it on, please. So if you look here for temperature, okay, you're also going to have to wear those plates. You're going to have three different plates with that medium called agar. And one of the plates is going to be kept at 4 degrees Celsius to also determine how that affects the rate of diffusion. And then, like I said, from those three plates that contain that gel-like substance called agar, one of them is going to have a higher percentage of agar. Therefore, the medium is going to be much denser than the other two to see how that affects diffusion. All right, I'm going to try again. Please check your microphones and turn off your mic, please, so we don't have any background noise. So if you look here for diffusion, you have to keep in mind if the membrane is present or the medium is present, okay, it's not enough, okay, to have a concentration gradient, an area of high solute concentration next to an area of low concentration. So yeah, I just don't have it on here, Chris. I'll find it real quick. Okay, so listen here. So what happens then? Yeah, I'm looking on it on here, and I can't find out who has theirs. Okay, they left. Never mind. All right, thanks, Chris, for reminding me. All right, so if you look here, if a membrane is present, okay, it's not enough to say diffusion is going to occur if we have a concentration gradient, okay? If the membrane is present, in other words, we can have an area of high concentration on one membrane, on one side of the membrane, and an area of low solute or no solute concentration on the other side, okay? But if a membrane is present, we have to ask a second question. Even if there is a concentration gradient, we have to ask, is the membrane permeable to the solute? Can the solute pass through the membrane? And this is going to be one of the questions that you're going to have to answer. For the first part of the experiment, you're going to be using two different dyes, and the molecules of dyes are different sizes, okay? So that alone can affect the rate of diffusion. For the third part of the experiment, okay, you're going to be working with glucose and starch. You have to know the difference between these two. They're both carbohydrates, but the difference is glucose is a monosaccharide. And if you take hundreds and hundreds, okay, of glucose molecules and covalently bond them together, you get one molecule of starch. to give you an idea of the difference in size. Okay, so the size can affect whether or not. So as I said, the lab procedure 1, okay, for lab procedure 1, it focuses on diffusion. Your medium, okay, is going to be your agar or agarose gel, one and the same, agar and agarose gel, one and the same, seaweed extract. That is your medium, or more specifically, your semi-permeable membrane. You're using 2 different dyes. They're showing you the formula weight, but that's the lab's way of telling you the molecules of dye that make up the methylene blue are much larger than the molecules of dye that make up the potassium permanganate. So for all 3 plates, you're going to have both dyes. So at a minimum, size is going to be an issue between the 2 dyes and how it affects the rate of diffusion. Okay. Now, whatchamacallit, the measurements for the rate of diffusion are taken every 15 minutes, and they're taken in millimeters, which I've already put in for a table for you. But then there's an issue of temperature. Okay, you have 3 different plates. It's going to talk about how plates A and C, what they have in common besides adding both types of dye to them, is that they're both made up of the same percentage of agar or agarose, which is 4%. Okay, that's A and C. The difference between plates A and C is that plate A is kept at room temp, roughly 22 degrees Celsius. Plate C is kept at 4 degrees Celsius in the fridge to see how temperature affects the rate of diffusion. So those are the 2 plates you want to compare if you're just strictly focusing on how temperature affects the rate of diffusion. However, plate B, it's the medium. That's the key. Now, A and B, plates A and B, what they have in common is both dyes are added to them, and they're both kept at room temperature. The difference is plate A is made up of 4% agarose gel, the medium, okay? Plate B is made up of 10% agar or agarose gel, meaning it has a much denser medium. So if you get a question about medium and how the medium affects the rate of diffusion, you would want to compare plates A and B. Now, so this is the information here, and the percentage of agarose, okay, and the differences in percentage of agarose as well as the percentage, it's listed on page 8 of your lab. So I'm giving you the page right here where to locate it. All right. Now it talks about plate C here, down here. Plate C, normally when we're in person, okay, should be out of the fridge as little as possible. So normally what happens when we do this in person is as you take the measurements of the diameter of the dye to determine the rate of diffusion, plate C, I would tell the students, go over to the little fridge we have by the sink, keep the plate near the fridge, measure the diameter in millimeters using a metric ruler, and then put it back in the fridge as quick as you can. Plates A and B, okay, they're kept at the same temperature. They have the same 2 dyes. The difference is the percentage of agar or agarose used to make the 2. The focus again of lab procedure 1 is diffusion and factors that affect diffusion. The second experiment in the lab procedures, part 2, focuses on osmosis. So you have to know osmosis is a special type of diffusion, and the way I want you to think of it is this way. I lied a little bit about diffusion. Diffusion is just the movement of molecules from a high to a low concentration, all right? But a lot of your textbooks, what they do is when they talk about diffusion, they always talk about the solute. So it leads to confusion for students. So what I do in this class is if I ask you about diffusion, we cheat, just focus on the solute. We'll leave out water because there's a special type of diffusion called osmosis. And this is where we focus on the movement of the solvent water. So just separate the 2 for now. Okay, let a A&P professor put the 2 back together. But if you're talking osmosis, listen carefully. If you're talking osmosis, this is the correct definition, 100% true. We focus on the movement of water. We focus on the movement of water across a semi-permeable membrane. And what's attracting the water molecules? The area of higher solute concentration. Now the molecules of water will move across the membrane, okay, from one solution to another until we reach equilibrium. In this class, if a membrane is present, unlike for solutes, for water, if a membrane is present, automatically assume the membrane is permeable to water. So please remember that, okay? In this class, the membrane may or may not be permeable to the solute, but if it's water, the membrane is always permeable to water. Water can always cross freely. So if you look here, it shows you we have a semi-permeable membrane right here. And on each side of the membrane, we have two sugar solutions, two glucose solutions, represented by the large beads. And you can just see here by eyeballing it, you have more molecules of sugar on this side than you have on this side. Now, the total volumes are the same, but you can't let total volume throw you off. When we're talking about concentration of solute, total volume in this case isn't actually the key factor to determining if two solutions have an equal concentration of solute. Okay. In lab 2, it talked about one way to explain the concentration of solute in a solution is a ratio. For every 1 molecule of solute, how many molecules of water do I have that make up a solution? And that's what I'm going to use in a bit. Okay. So if you look here, they're showing you we have two solutions at different concentrations, but they're both glucose solutions. But we have a higher concentration of glucose on this side than this side. So let's take a step back. Based on diffusion, the molecules of glucose, since we have a higher concentration on this side of the membrane than this side, we have a concentration gradient. So the molecules of glucose do want to diffuse from a high concentration across the membrane to a low concentration. But notice the pore sizes in the membrane compared to these molecules of glucose. The glucose is too large. So this is more indicative of what happens with our cells. So what happens is, even though there's a concentration gradient, diffusion of glucose is inhibited. It doesn't happen because the glucose can't move through the membrane. The membrane is not permeable to the solid glucose. However, as I said, we always assume that the membrane is permeable to water. And what happens is, while diffusion wants to occur but the membrane is inhibiting it, osmosis also wants to occur. We focus on the movement of water across a semi-permeable membrane. So water starts moving across the membrane. Now, water is actually moving both ways. But since the higher solute concentration is on this side, what happens is more molecules of water move from your left to your right than in the opposite direction. Therefore, movement of water is both ways, but net movement of water is from our left in the direction of the higher solute concentration to our right. And water will continue to move across that semi-permeable membrane towards the higher solute concentration until we reach what? Equilibrium. Ignore this, I forgot to cut this out. This one here, if you look at this one here, okay, notice up here it says at equilibrium. Water has moved across the membrane towards the higher solute concentration until we reach equilibrium. The arrows indicate water still moving both ways at equilibrium, okay, but there's no net movement of water anymore. There's not more molecules of water moving from your left to your right or your right to your left. These two solutions are at equilibrium, meaning they have the same concentration of solute. But what throws students off is that the total volumes are not the same. That doesn't matter. You go back to lab 2 and it talked about how you can use ratios to explain solute concentration. So I'm going to use a hypothetical situation here to explain solute concentration comparisons between these two. Let's say on this side, okay, of the membrane, for every 1 molecule of glucose that I count 50 molecules of water. That's the ratio. For every 1 molecule of glucose I have 50 molecules of water. Let's say I come over here to this solution on the other side of the membrane, okay, and I count up the number of sugar molecules to water molecules, and I come up with the same ratio. On this side of the membrane, for every 1 molecule of sugar I count, I have 50 water molecules. So the ratios are the same, 1 to 50. That means they are at equilibrium. They are at equal concentrations. So you can't go by total volume, everybody, okay? You have to go by the number of molecules of solute per number of molecules of solution or water if it helps. Some of these slides, I'm trying to help you out. Okay, so when you're doing your pre-lab, look at these slides. Some of the slides I have numbered, and these numbers correspond with pre-lab questions. So I'm going to let you look at those. Is that clear to everybody? Okay, I'm going to let y'all look at those. And then if you notice these next 2 slides, 14 and 15, I put page 4. That's to help you with the pre-lab at the top of page 4. So I've given you some help here answering the questions on there, but you're still going to have to go over it and study it, okay? I'll let y'all look at that. Tonicity. Whenever you hear talk about tonicity, all right, automatically tell yourself it has to do with osmosis and the movement of water, and how the movement of water can affect water balance in a cell. Okay, so in lecture I said the way I describe or define tonicity is that it has to do with how the movement of water across the membrane of a cell creates pressure called osmotic pressure, and that this movement of water across the membrane of a cell could affect the shape of a cell, causing a cell to swell or to shrink. Therefore, we're talking water balance here. Now, what promotes the movement of the water across the membrane, thus affecting the tone of the cell or the shape of the cell? It has to do with the solute concentration in the ECF. So these three right here, these are the names of solutions that have an effect on water balance in a cell. But the names of these three solutions are applied to your ECF, your extracellular fluid. Okay? So in part 2, what the students would normally do is they would take this, it looks like a plastic-like or sandwich bag-like material, but it's in a rectangular shape, and the students would fold up one end and put a clamp on there, and on the other end they would open up and fill it up with a particular solution, and I'll show you what solutions. Then they would clamp the other side, and they would take what is called dialysis tubing and turn it into a little bag filled with the solution. And that little dialysis bag filled with solution would function as your cell. The material that makes the dialysis bag, that functions as your semi-permeable membrane. The solution inside of the bag is ICF. And then what you would do is put the bag inside of a beaker filled with the solution. Depending on the makeup of the solution in the beaker, okay, that solution in the beaker could be behaving as an isotonic solution, a hypotonic solution, or a hypertonic solution. But in either case, that solution in the beaker represents your ECF. Okay, so for tonicity, solutions surrounding cells exerting pressure on cells called osmotic pressure. This is because solutions have different concentrations of solutes compared to the solute concentrations within cells. So we're talking the solute concentration of the ECF versus the ICF. Therefore, for the solutions outside of the cell, the ECF can be described as either being an isotonic, hypotonic, or hypertonic solution. So this is the case here. This is the same figure that I used for chapter 7, and I'm going to repeat the same thing I did for the lecture of chapter 7. When I was taking physiology, we had a lab where we studied osmosis, specifically tonicity. All right. And what would happen is the professor would give us 3 test tubes. One of the test tubes, and I don't remember the exact concentration, but one of the test tubes would have an ideal salt concentration that did not affect the cell shape. Therefore, that solution in the test tube was an isotonic solution. It was our ECF. Somebody in the group had to prick their finger, put a couple of drops of blood to introduce red blood cells to the test tube, and the red blood cells would maintain a normal shape, what we call a biconcave shape. Because the concentration of solute in the solution in the test tube was at equilibrium with the concentration of solute within the red blood cells. So water balance was ideal. Water was moving in and out of these cells, but the cells weren't gaining excess water and they weren't losing excess water because they were at equilibrium. Therefore, the ECF is said to be an isotonic solution. But in 2 other test tubes that would be given to us, one of the test tubes just had water in it. That was it. So whoever was the one donating the blood, they would put a couple of drops of blood into the second test tube that had just water. Well, that water in the test tube was functioning as our ECF. And since it was just water, obviously the solute concentration inside of the red blood cells introduced to that water had a higher solute concentration than the water in the test tube. Therefore, water would move across the semi-permeable membrane of the red blood cell into the red blood cell and would continue to move into the red blood cell trying to reach equilibrium. In other words, moving into the red blood cell until the ICF and ECF were at equilibrium. The problem is it was just water in the test tube. So eventually what happened is the cell would gain so much water, it would swell, and some of them would even burst. The membrane would be placed on such a great amount of stress, it would physically break, and the cell would lyse or burst open. In this case, whenever the ECF caused the cell, okay, to have an imbalance in its water balance and to swell because it takes on excess water, we say the ECF is behaving as a hypotonic solution. Then the professor would give us another test tube. And this one, again, I don't remember the exact concentration, but this test tube would have a high salt concentration, okay, in comparison to the solid concentration in the cell. So again, the student donating the blood would put a couple of drops of blood into this test tube filled with the salt or saline solution that it was at a high concentration of solute. And what would happen now is, since the solute concentration of that saline or salt solution in the test tube, which was much higher than what was in the cell, water in the cell would diffuse or would move across the cell membrane in the direction of the higher solute concentration. But our professor would make sure that the saline or salt concentration or the solute concentration was so high that so much water would move out of the cell in the direction of this higher solute concentration, trying to reach equilibrium between the ICF and ECF, the cells would shrink or shrivel. In other words, the cells would become dehydrated. Whenever the ECF causes a cell to lose water, maybe to the point of shrinking or shriveling, we say the ECF is behaving as a hypertonic solution. Well, in the second lab procedure, second part of the experiment, the lab procedures, what you're focusing on initially is osmosis. Okay, there are two solids you're going to be working with, glucose and starch. Okay, and then the solvent is always water. The material you're going to be working with, it's a dialysis tubing, as I said, that you would turn into a dialysis bag filled with a solution, and this dialysis bag with the solution inside of it would function as your cell. This figure here, I believe it comes off of page 1 or 2 of your lab protocol, but I moved it. Okay, the reason why I moved it is because, listen carefully, the last set of pre-lab questions, you need this figure, slide 18, to help you answer the last set of pre-lab questions. It just so happens the last set of pre-lab questions, as well as this slide, okay, are related with experiments 2, or part 2 and part 3. So please remember that, okay? This slide will help you with the last set of pre-lab questions, and then that last set of pre-lab questions along with this slide should help you with part 2 and part 3 of your lab procedures. That dialysis tubing, okay, when you work with it in person, it looks pretty solid, but it's actually a semi-permeable membrane. It's definitely permeable to water. That's why I have the arrows going both ways. And it's even permeable to glucose. But if you recall, you have to know what starch is. It's a carbohydrate like glucose, but it's not a monosaccharide, it's a polysaccharide. Okay? Starch is formed when plants take hundreds upon hundreds of molecules of glucose and covalently bond them one to the other, creating starch. So a molecule of starch is much, much larger than a single molecule of glucose, which is much, much larger than a single molecule of water. However, it tells you in the protocol the dialysis tubing is permeable to these two. Therefore, I show arrows going this way. But the starch, the dialysis tubing, does not contain pore sizes large enough for the starch to move through. Okay, so that's to help you out a little bit there. This is part 2 for your lab experiment. This is how you would do your setup. Okay, now you're not actually going to do it in person, but there is a video link, okay, created by one of our professors. Make sure you watch the video. He goes over the setup. He didn't set up all 4 samples. I wish he had, but he didn't, but I ain't going to complain because he did it, not me. But he does go over it and he narrates what you would have done, as well as showing you the procedure, showing you everything from part 1, what the Petri dishes look with that medium called agar, to part 2 with the dialysis tubing, to part 3, okay? So make sure you watch the video link I provided. Had we met in person, I would have told you, hey, you have 4 beakers on your tray with a piece of tape, just label them number 1, 2, 3, and 4. And then I would have told you, hey, look, add 150 mils of the appropriate solution to each beaker. First 3 beakers, you would have added 150 mils of water to each beaker. The last beaker, you would have added a 10% glucose solution. What does 10% glucose mean? 10 parts of the solution is made up of glucose. The remaining 90 parts, or the remaining 90%, is made up of water. It's a solution. So that automatically should tell you the solvent is water, and if only 10% is glucose, the remaining 90% is water. So you're not always going to be shown water's there, but it's supposed to be understood because it's a solution. Then we talk about the dialysis tubes that eventually you would fill with the solution and have clamps on both sides, transforming into dialysis bags, which would function as your cells. Okay, the dialysis bag serves as your semi-permeable membrane. The solution on the inside is ICF. So if it helps any, what you put in the beaker, these solutions are your ECF. What you put inside of the dialysis bag, that is your ICF. Okay, that is your ICF. Now, if you notice here for the dialysis bags, okay, for the first dialysis bag, you're only adding a 1% starch solution. That's not a high solute concentration. It's a very low solute concentration. So this is the catch when it comes to osmosis and tonicity. If there's not a significant difference between the solute concentration in the ECF and the solute concentration in the ICF, okay, water will move across the membrane of the dialysis bag, but since the solute concentration of starch in the bag is only 1% and it's not a high percentage, you may or may not get net movement of water from the ECF across the dialysis bag membrane into the ICF. You may get a little bit or none at all. So that's a catch, okay, as opposed to these two down here. Notice bag 2, the ICF is going to consist of 20% glucose, but bag 3 is going to consist of 40% glucose. Now what you have are two different ICF solutions in the dialysis bag, which have a significantly higher solute concentration than what is in the ECF, which is just water. So for these two bags, if you know the definition for osmosis, you would expect water to diffuse across the membrane of the bag into the ICF where there's a higher concentration of solute. And you would expect water to continue to move into the bag until the two solutions reach equilibrium. Well, the reason why we do 20% glucose to 40% glucose is to demonstrate that the greater the difference in solute concentration between the ECF and the ICF, the greater the amount of water, okay, that will move towards that higher solute concentration. Okay, now if you look at these two here, the ECF is going to be made up of 10%, the ICF is going to be made up of 10% also. So if you notice, the concentration of solute in the ICF is the same concentration of solute as in the ECF. Therefore, these two solutions are simply at equilibrium. So for this last one here, if I were to ask you, based on tonicity, since these two solutions are at equilibrium, how could I label this ECF in terms of hypotonic, hypertonic, or isotonic? I would say in this case, since the ECF has a solute concentration that's already equal to the solute concentration in the ICF, I would say it's an isotonic solution. That's how it's functioning. But if you look at these other three, just going by numbers, they're all water. The higher solute concentration is within the dialysis bag in the ICF. Therefore, whenever the ECF has a lower solute concentration than what's in the ICF, we say the ECF is functioning or behaving as hypotonic, especially in this case for beaker 2 and 3 versus bag 2 and 3, because it's significantly different. So keep that in mind. Now listen, for both lab procedure part 1 and lab procedure part 2, I've given you data in the table. Okay. For lab procedure part 1, I've given you measurements in millimeters. Take a look at them first before you start answering the post-lab questions so you get your mind oriented properly. And then in the second table, I've given you the weights of the dialysis bag. Before we put them in the, giving you 2 weights for the dialysis, these 4 dialysis bags. The weight of the dialysis bag before and after we place them in these ECF solutions. Okay. The way we determine if osmosis occurred is by measuring the weight. That's why I've given you an initial weight and a final weight. Okay. I've also given, I believe, hopefully I remembered, I gave you a time of 1 hour. Okay. The dialysis bags would have sat in the beaker solution or the ECF for 1 hour before you weigh them a second time. And the way it works is the reason for weighing the bags before and after you put them in the beaker solution is because by detecting if a change in weight occurs, that's how we determine if osmosis occurred experimentally. It's by a change in weight. Except, as I said, if you can predict how the ECF is functioning, hypotonic, hypertonic, or isotonic, you not only can predict if you would expect a change in the weight of the dialysis bag, but you could also predict whether you expected that change in weight to increase or decrease, depending on the movement of water. Okay. Now to backtrack a little bit, and I'm sorry, but to backtrack a little bit, the way you're determining if diffusion occurs, I need to clarify this. The way you're determining if diffusion occurs, okay, is if you look at table 1 in the lab procedure, it was by measuring the diameter. That's why I put this little line, by measuring the diameter of the dye. So as I said, there's a video watch. He sets up the experiment. He puts the drop of dyes in this medium called agar or agarose gel, and then he'll take a measurement of the diameter, and then every 15 minutes he'll measure it again and again. Okay, measuring the diameter of the dye every 15 minutes is how we're determining if diffusion occurs. So I'm sorry I forgot to point that out. All right, now there's 3 parts to the experiment. Your setup here for part 2 of the lab procedures is also going to be used for part 3 of the lab procedures. Part 3 of the lab procedures basically is saying, hey, look, I know in part 2 you determine if osmosis occurred by measuring a change in the weight of the bag, because any change in the weight of the bag was going to be due to the movement of water into or out of the bag. Part 3, though, wants you to try and determine, is it possible for not only water to be moving towards the higher solute concentration, but if the membrane is permeable, is it possible for the solute at the same time to be moving from a high concentration of solute to a low? In other words, the water molecules are moving in the opposite direction than the molecules of solute. So for part 3, you're only going to focus on the first 3 beakers. We don't focus on the 4th beaker solution because what's in the bag is the same concentration of solute as what's in the beaker. We focus on these first 3 beakers for part 3 to determine if in this same experiment we set up for osmosis, did diffusion happen? Did starch and glucose diffuse out of the dialysis bag into the beaker where we only had water? High concentration was here in the ICF. Did they diffuse out into the water where there was no solute? This is where I said you need to use that figure. We talked about the size of starch in the dialysis bag. So the way we would do this last part of the experiment, and again, this will be on the video, so watch the video, okay, is there are 2 chemicals we normally use in the lab in person. These are what I call sometimes chemical color indicators. Okay, the 2 chemicals are Benedict's reagent and Lugol's iodide. Benedict's reagent is a chemical that is used to detect if glucose is in a solution. The catch is this. The way it works is if you want to determine if glucose is present in a solution, you'll take a little bit of that solution and put it in a test tube. You'll add a few drops of Benedict's reagent. Benedict's has a natural blue color. So if your solution is clear, when you add the Benedict's reagent, it'll turn your solution blue. The catch is this. The Benedict's reagent will not react with the glucose unless you expose it to boiling temperatures for roughly 1 to 2 minutes. Okay? So what happens is students will take some of their beaker solutions, okay, out of beakers 1, 2, and 3, and add it to 3 test tubes appropriately labeled 1, 2, and 3. They'll take about a ml of solution out of the beaker, okay, after an hour. And then what they'll do is they will add Benedict's reagent to the test tubes, especially the ones that you're trying to determine if glucose diffused out of the dialysis bags. It'll turn the color of the solution from the beaker blue, and then you place it in boiling water. And if glucose did diffuse out of the dialysis bag, what happens is your sample in the test tube goes from a blue color to a yellow-orange color. It's a very cloudy color, and then depending on how much glucose is present, it could be more yellow or more orange. But in either case, If when exposed to boiling temp, the Benedict's reagent detects glucose, it'll react with it, change the color of your solution, you know it's positive. However, if after 1 to 2 minutes your solution stays blue, then you know glucose isn't present, it's negative. So you're going to use this chemical, but you test the beaker solution, so read the procedures carefully. For starch, in person, it's a much quicker experiment. There's no boiling involved. If we want to know if starch diffused, okay, out of the dialysis bag into the beaker of water for number 1, all we need to do is take some of that water from that beaker number 1, put it in a test tube, roughly 1 ml, and then we add a few drops of this chemical, Lugol's iodine. Lugol's iodine has naturally a yellow color to it, and if you add it to a solution that's clear, it'll turn the solution it'll turn the solution yellow immediately if the solution is negative for starch, if there's no starch. So there's no boiling involved. However, if you transfer some of the solution to the beaker into a test tube, and then you add a few drops of Lugol's iodine, but the solution suddenly turns a blue or a blue-black color, then that means the Lugol's iodine detected starch, reacted with the starch, turning your solution a blue or black color, meaning the solution is positive for starch. But keep in mind, I gave you that slide. Okay, look at that slide, and I'm giving you the information as to whether the dialysis tubing is permeable or not. Now, in the lab procedures, okay, there's a couple of spots there where they talk to you about labeling tubes B1 through B3 and L1 through L3. See if you can fill out the table based on what I gave you. You don't have to turn it in, but it just helps give you a practice run. And then the very last table, which is under part 3 of the lab procedure, that's the table you definitely have to fill in to help you answer your post-lab questions. Okay, so if you look here at your pre-lab, there are a lot of questions and a lot of answers. Listen carefully. If the questions are multiple choice, don't just give me the answer, give me the letter so I can speed up the grading process. I get students that say, well, why aren't you grading faster? Well, talk to your classmates or talk to yourself, okay? Keep the format real good. The majority of you are doing a good job, okay? So that just helps me speed things up. Post-lab, you're going to do parts 1, 2, and 3. We do not do, okay, we do not, do the experiment having to do with the Elodea leaf and the cells. So in your lab protocol, I believe at the beginning under lab procedures, where I give you the video link where the professor demonstrates what you would have done in person, below that video link, I put down omit Elodea leaf. So when he reaches towards the end of the video, when he reaches the point of pulling out the microscope and talking about the Elodea leaf, you don't have to watch that last part, okay? So there's a part of the video at the end you don't have to watch. You're just going to answer parts 1, 2, and 3. I removed the questions from the Elodea leaf. You're going to create some graphs, so make sure you look at how you're supposed to label those graphs. Again, tutoring does help you out. Okay, our tutors are supposed to be able to help you out, including creating the graphs, all right? So that's part of their job. That's part of their tutoring on there. But I also posted those tutorials on there, those documents, the PDF documents. Now let me go and pull up the file itself. Okay. So let me jump ahead here. I'm trying to jump ahead. Come on. Sorry, everybody. It's not cooperating right now. Page 2 is where you have that figure that I'm trying to get to. Page 2 is where you have that figure. Okay, we're freezing up here, so y'all may have to just take some of this. Page 2 is that figure where it shows you the little cylinder which represents the dialysis tubing, and it shows you starch, glucose, and water. But I moved it in the PowerPoints, okay, to go with your last, to help it better match up with your pre-labs. So everything I want to show you, I'm not going to be able to show you. I can scroll down on the side. I want it to go to the pre-lab. Okay, let's see if we get lucky here. One of the pre-labs on here, I don't know if it's page 4 or 5, but it's a pre-lab that gives you the polarity of, I believe it's glucose and fructose on 2 sides of a semi-permeable membrane. Okay, and the questions are basically, is there movement of glucose, or here we go. The questions are basically, is there movement of glucose, movement of fructose, or movement of water across the semi-permeable membrane? Okay, you got to read the information up here to know whether to answer it, but if it's asking you about only glucose, ignore the fructose. If it's asking you, okay, only about fructose, you can ignore the glucose. If it's asking you about the movement of water, simplify it and just add up the molarities. Just do simple math and add it up just to simplify it for yourself, because I'm more interested in the process. It'll tell you about the membrane and whether or not it's permeable. But it's going to ask you two questions, and it throws students off. It'll say, is there movement of, say, glucose across the membrane? Then the very next question will be, is there net movement? All right. If they're talking about movement versus net movement, I'm going to draw this not to any particular order. If they're asking you for movement, let's say it's water. Okay. Water can definitely cross this membrane. So if they're asking you if there's movement, you would say yes. Movement meaning water's moving both ways. If they're asking you about net movement, then in that case, you have to figure out if on one side of the membrane or the other, is there a higher solute concentration? And if there is, I'm not going to do this, but if there is, then you would draw in extra arrows pointing in one direction more than the other to symbolize net movement. Is that clear to everybody? So keep in mind, if one side of the membrane or the other has a higher concentration of solute, at the very least, water could move across the membrane, and not only move across the membrane, but there will be net movement. More water will move one way than the other. Likewise, if two solutions, which it's going to ask you this on the next page, if two solutions are at equilibrium, okay, they have equal concentrations. Well, if the two solutions are at equilibrium, there's no net movement of water. However, is water still moving both ways across the membrane? Yes. Okay, so keep that in mind, please. And then this is the one I was telling you about. I moved that PowerPoint with the dialysis tubing and then the starch and the glucose and the water and the little pictures and the arrows going both ways. And for starch, I put an X. Okay, this is where you'll need to use that figure here. And then again, starting with lab procedure 2. All right, on here. Are there any questions? Okay. Yeah, what's your question, Guadalupe? You can turn on your mic if it's faster.
Yes. So for the net movement of water, would be going both directions, right? Because
None of it's
no,
net
wait.
movement. Movement of
Oh,
water, you would indicate if you say movement of water is occurring, then it's occurring in both directions.
okay.
But if it's net movement of water, not only is water moving in both directions, but there are more water molecules moving in one direction or the other. Let me just listen carefully. This isn't the scale, okay? So ignore the concentrations. Ignore the concentrations that are here. Okay, let me do this again. If it asks you if there's movement of water, well, any membrane that's used in this lab, you're automatically tell yourself it is permeable to water. That means water can move across. So movement of water would be indicated this way. There should always be movement of water, okay? But if you're asked about net movement of water, then the way I would represent it using this figure, ignore the concentrations. I'm just doing this arbitrarily. If I wanted to indicate there was net movement of water, then I would draw more arrows pointing in one direction than in the other direction. Does that make sense there, Guadalupe?
Yes, it does. Yes, thank you.
Okay. So keep in mind, when two solutions are at equilibrium and separated by a membrane, there is movement of water, but there's no what? Net movement. Why? Because the solute concentration is the same. Okay? Now, what you're going to end up doing, say, with this figure, okay, is that the arrows, which way most of the arrows are pointing, if the solute concentrations are not equal, which way the arrows are pointing will be pointing in the side or towards the side that there's a higher solute concentration. Does that help? Because I'm tripping over my tongue right now.
Yes, it does.
Okay, all right. Any other questions? Listen, everybody, you have to study. I mean, there's no two ways around it. And watch the videos. Lab is normally in the summer, lab's 2 hours and 40 minutes, and everybody works the entire lab, okay? And that doesn't include the lab report. You've got to watch the videos and go through everything. We do have some students that are doing an amazing job with their lab reports. Everybody else has got to step it up, jump on top of things real quick, okay? Any other questions? Okay, then I'm going to stop the recording.